Everyone, this is Dr. Pradibha from Department of Biotechnology, Kamaraj College of Engineering and Technology. Today we will look into a very important aspect of structure-function relationship of enzymes. So among it we will choose serine protease as an example, and we will discuss and delve deep into how the structure of the protein is enabling its function to happen. So proteases, what are proteases? These are a class of enzymes which are involved in cleaving the peptide bond in proteins, and they are commonly found, or they find their use in food digestion, cleavage of precursors, signal peptides, and these are some of the biological functions that proteases do in the body. Classification of proteases is based on the important amino acid residue that is present in the active site of the enzyme. So based on it, it can be classified as serine proteases, cysteine proteases, aspartic proteases and metalloproteases. Common examples of serine proteases include chymotrypsin and certolizin. Metalloproteases include matrix metalloproteinases and all that. So as I told you, today we will look into the structure function relationship. So we will have to learn about the structure and the function. So first we will look into the function of enzymes. Enzymes, as you know, are biological catalysts which tend to enhance the rate of any biological reaction. The mechanism by which it is happening is the transition state intermediate is stabilized and the activation energy of the overall reaction is reduced. So how does the enzyme do that is what is we have to see in today's lecture. So enzymes decrease the activation energy of a chemical reaction that it catalyzes, and you can see this is expressed by the Michael-Menten equation, that is the saturation kinetics of enzyme. So going specific into the function of serine proteases, as I told you, they cleave a peptide bond, that is formed between two amino acids. So it catalyzes the hydrolysis of peptide bonds within the polypeptide chain. The bond that is cleaved is called a sessile bond. Serine proteases cleave the peptide bond by forming a tetrahedral transition state intermediate. So as shown in the picture here, this is a polypeptide chain with amino terminus and carboxy terminus over here, and this is the amino acid chain, and this particular CO that's missing here, CO-NH bond is the sessile bond. The bond between C and N is the one that's going to be cleaved. So after the enzyme functions, you can see that two different peptides are formed because the cut is made at the specific sessile bond. This takes place in two steps. In step 1, there is a reactive serine amino acid that is responsible for doing this, and there are actually amino acids that are importantly present in the active site which are responsible for the function of the particular enzyme. One among that amino acid is serine. So serine residue in the active site, you can see this is a serine residue with its side chain, that is CH2OH, which normally is bound to a histidine that acts as a base capturing this reactive OH group. So normally the enzyme in its resting state is like this, and during this step the C1 of the substrate acts as joins in a tetrahedral geometry to form a transition state. You can see this is a peptide bond, this is the peptide substrate. Enzyme cleaves the peptide, okay. So this is a peptide substrate and this is the sessile bond that is mentioned here, and a tetrahedral transition state intermediate is formed between the serine residue and the substrate by this OH group. So this is called the acyl enzyme intermediate, and by the end of step 1 you can see the bond is cleaved and one part of the peptide is released. In the second step, the other part of the peptide also has to be released, and so this also happens through another acyl enzyme intermediate, but here it is a deacylation process involving a water molecule in this enzymatic reaction. So here you can see that the serine is now covalently linked to one part of the peptide which has to be cleaved, and a tetrahedral transition state intermediate involving base histidine and the serine residue. Now the water molecule, the hydrogen of water molecule is reconstituted to histidine, and OH reconstitute the COOH bond of the outgoing peptide. So that is how the second peptide also leaves the enzyme. So now that we have seen how the functioning of the enzyme is taking place, we now have to relate how the structure of the enzyme is responsible for doing this function. So, structure function relationship, as you can see, these areas which are shaded in grey are parts of the enzyme, and those which are shaded in pink are parts of the substrate. So, this substrate, which is again another protein because it is a protease, this substrate has to bind or interact with different parts of the enzyme for this specific function to happen. So, what is the specific function? Formation of an acyl enzyme intermediate and cleavage of peptide bond. So, to do that, the enzyme need to be spatially oriented such that all these contacts are made at the right point, because all these peptide bonds are there across the main chain, but only the sessile bond has to be cleaved. So, in order for the enzyme to recognize what is the sessile bond or where exactly is the sessile bond, these interactions are very essential. So, what are those interactions? There are 4 important structural features of an enzyme that are required to perform this function. First one is called the catalytic triad. Triad means 3, as you know. So, here the catalytic site has 3 amino acids that are very important. Number 1 is an aspartate, 2 is a histidine, and 3 is the reactive serine, which we saw earlier. So, these 3 amino acids are very important for the functioning of the particular enzyme. So, histidine acts as a general base, and aspartate and serine, all these 3 enzymes are important as a catalytic triad. Second one is a oxyanion hole. Because the tetrahedral transition state has to be stabilized, this oxyanion hole gives a kind of a correct electrostatic environment for the stabilization of oxygen atom, and that is how this tetrahedral transition state is stabilized by the enzyme. Third one is main chain substrate binding. This is actually non-specific binding, where this is the substrate's main chain and this is our enzyme's main chain, and they interact irrespective of whatever the R group that is present in the substrate. The main chain interacts, thereby aligning the substrate along with the enzyme exactly for the sessile bond to be cut. The fourth feature is a specificity pocket, which is truly responsible for recognition of the sessile bond. Say for example, some enzymes cut after a hydrophobic residue, some cut after a positively charged residue. So, these are recognized by the specificity pocket, and they define the sessile bond in any protein that has to be cut. So, for chymotrypsin, it always cuts after a very large aromatic amino acid like phenylalanine or so on, and for trypsin it is always after lysine or arginine. So, you can see that there is a specificity pocket, oxyanion hole, the catalytic triad, and the main chain substrate binding interactions, which are essential for protein function. Now, we are going to relate how the structure of chymotrypsin is going to do this function, and we are going to fit all these 4 features of the enzyme that are necessary for the function into the structure of this enzyme. Chymotrypsin is basically a 2-domain enzyme, or it has 2 antiparallel beta barrel domains, and each domain has around approximately 120 amino acids formed by a simple Greek key motif and a beta hairpin. So, this is the overall structure of a chymotrypsin enzyme, and the active site is actually in between, sandwiched between the 2 domains. At the phase of interaction of the 2 domains, we can see or locate the active site. And these are the 2 domains in a beta barrel representation. We can see that serine, histidine and aspartic acid. Aspartic acid and histidine are part of the second domain, whereas serine is part of the first domain, and all of the 3 come together in 3-dimensional structure, forming the catalytic triad. This is the site of main chain substrate binding. This blue region you can see is main chain substrate binding region, and this is the specificity pocket where the substrate the R group of the sessile bond can go and fit into. So, this is how the 3-dimensional structure of a protein enhances it to do its function. So, in conclusion, we have seen that the chymotrypsin is an enzyme that cleaves a peptide bond specifically after a specific residue, and this is how the structure of chymotrypsin is helping it to do its function. As far as engineering these proteins are concerned, this specificity pocket can be engineered to adapt to accommodate different kinds of substrate, thereby expanding its number of substrates that can be used by the protein. So, if for example, you can see that a serine residue at the base of this pocket and two glycine residues on the sides of this specificity pocket, this allows a phenylalanine, a bulky residue, to enter into the pocket. Assume that instead of serine there had been an aspartic acid. So, aspartic acid, as we know, is a charged amino acid and it cannot actually accommodate a big bulky amino acid. Instead, this negatively charged aspartate is willing to accept a positively charged lysine. So, this case the specificity pocket can be specific for a lysine in the sessile bond. Next is an enzyme choleelastase, where you can see that instead of glycine lining the pockets, there are valine and threonine. Valine is a hydrophobic amino acid, as we know, and this does not permit long bulky amino acids or charged amino acids to enter into the specificity pocket, allowing only small amino acids like glycine or other aliphatic amino acids to be their substrate. So, if possible, we can engineer the active site of an enzyme to expand its substrate selectivity. Okay. So, understanding proteins structure function can help us to engineer them in a good way. Thank you so much for your patient listening.